So we're here right at the end of Chapter 1, and we're just going to finish up Chapter 1 real quick. We already talked about measles and how it can progress to SSPE pretty quickly, and so that, in my mind, is one of the big reasons, a big explanation that you all, now as trained microbiologists, should tell your friends that have concerns about vaccinations. If their young child gets measles and then by 20, 25 years of age progresses on to SSPE and ends up dying from that disease, then that's much worse than most of the even perceived complications that some people imagine might arise from immunizations. But we have had a lot of effective immunization campaigns, and we've talked through some of those as we went through earlier parts of this chapter. One of the reasons that immunizations are effective is because of what's depicted in this particular graph. If you've never been exposed to a specific pathogen and the antigens that it produces, the first time that pathogen gets into your system, it has between a week and 10 days of free reign. The only thing that can impact it at that point, unless there's some natural antibodies circulating, are components of the innate immune system. So those phagocytic cells that we talked about and some other cells might be able to deal with that, but that's all that can deal with it for about 7 to 10 days. Because adaptive immunity takes that long to kick in. Because adaptive immunity requires that cells already in existence in your body mutate and change and be selected for by the specific antigens that pathogen is bringing in. That's part of the clonal selection that I just asked you about on the quiz and the clonal expansion. One or a few cells will get selected by the antigen, and then they'll go through the amplification process of clonal expansion, but that takes time. So it takes time for them to respond because, one, the cells have to be produced in your body. They're not in existence if you've never been exposed to that particular antigen. And they're not very effective if they are in your body. So when we administer a vaccine or anything that's going to give you immunity, at day 0, the amount of antibody that's circulating in the bloodstream is very, very low. And that's what we're looking at here. The y-axis is antibody concentration in the serum or circulating in the bloodstream. And so this is when you came in contact with that antigen. If it was a virus or a bacterium that you hadn't seen before, it'd be the same thing. And you can see by day 8, there's still not much response. But day 7, 8, you begin to mount an adaptive response and begin to produce some antibodies that are specific for that pathogen. And then by day 10, 12, 16, usually around day 10, it plateaus and it'll maintain that concentration in the serum, and then the concentration in the serum will drop off. There's specific antibody classes that are produced during that process, and we'll talk about some of those down the road, but that gives you an idea. If you look at the serum concentrations, there's not a whole lot on the microgram per milliliter level of plasma serum antibodies that are there, even after the first exposure. If you compare that to some of the concentrations that you should have already seen in Chapter 2 as you read through, and as we'll look at when we go through for complement proteins, these are very, very low concentrations that are produced here, less than 10 micrograms per milliliter after the first exposure. But if we make a secondary exposure, at that point with the first exposure, not only are plasma cells produced that can make antibodies specific for that antigen, but some cells called memory cells are also produced, and they persist in the body. We mentioned that last week just a little bit. So those memory cells are produced during this process, and then later if you give a vaccine that has antigen A in it, the response that you get at that point will be much stronger. Look at this response for antigen A. It's up here 10,000 times stronger, or at least 1,000 times stronger than what you had on the first exposure to that specific antigen. That's because the cells have already been produced. Now all that has to happen is they have to expand up and make the antibodies that are appropriate for that specific antigen. And that process is much quicker than the mutational processes that have to happen to actually produce those cells that are going to give us this primary response. So the secondary response happens usually within 24, 48 hours on exposure to that same antigen. And we get a much stronger, much more specific response because of that. But they just throw in this for a twist. If at the same time you throw in antigen B into that mix, so now there's a new antigen that the body has never seen before, it's going to go through a primary level response. It's not going to have the same magnitude of response, and it's going to be much slower. It, again, is going to be 7, 8, 10 days before you begin to see accumulation of antibodies to that specific antigen. Does that make sense? It all happens because the cells that need to respond are not in your body right now, or they are very few in your body right now. And you're going to find out when those cells that can recognize a specific antigen do so, they also mutate. They also change, and some of them mutate into cells that can bind much more strongly than the original cells could. Others mutate away and they can't bind anymore, but those that bind the antigen strongly are the ones that are selected for in that process of clonal selection. So this is why vaccines tend to work. If you've been vaccinated, usually most vaccines have a course of 2 or 3 or some of them 5 different booster shots that you get along the way to produce that secondary response. This is called a primary response. This is called a secondary response. And once you have that secondary response, you've got lots of specific antibodies circulating in your bloodstream. And those circulating antibodies, as soon as that pathogen that's real comes into your system, can bind onto the virus if it's a virus, or can bind onto the antigens that are on the surface of the bacterial cell. And again, those antibodies flag the organism or infectious agent and help remove it from the system. So that's the process and how basically the primary, secondary immune responses work and why vaccines can be effective. Most of you have, or many of you probably have been immunized against the influenza. And so some of you are probably asking, why do I have to be immunized again every year? That guy, influenza type A, its genome is RNA. And it makes a specific RNA polymerase that doesn't do proofreading. Remember what proofreading is related to DNA polymerase? What is it? Somebody just tell me quickly. It's like a spell check, exactly. DNA polymerase, all polymerases can make mistakes, all enzymes can make mistakes, and when DNA polymerase makes a mistake, the main DNA polymerase that replicates the chromosomes can actually see the last little bit that it wrote, and if it made a mistake, it can erase or nibble away the end that it just synthesized as it's making the DNA strand. And if there's a mismatch there, it can chew backward, remove that mismatch, and then start over again. That's proofreading. Well, the RNA polymerase of influenza A doesn't have proofreading. That means as it's making copies of its RNA genome, it makes a lot of mistakes. In other words, it introduces a lot of mutations in the copies that are being made. So as it replicates, you're getting hundreds or thousands of different strains that are emerging out of one individual cell as those mutations are independently happening as the viral genome is being replicated. And sometimes those mutations are in the proteins that are the genes that code for the proteins out on the surface of the virus. And if they change the particular site on those proteins that the antibody was specific for, that antibody can no longer bind to that particular protein. And so the immunity that you produced against the strains that were out there last year may not work against the strains that come out this year. But what we do is try to look at the strains that were prevalent toward the end of the previous season and then make a vaccine with those strains, hoping that those same strains will reemerge, or strains that are very similar to those strains will reemerge, and so the vaccine will work. You probably know last year only about 50% of the people that got the influenza A immunization were protected. The other 50% got the flu because the strains that came out were mutated strains, changed strains, and it can be a mutation that changes a single amino acid that's right there where the antibody needs to bind. The antibody will no longer be able to bind to that specific protein, that particular protein that's produced. So that's also why we get all kinds of emerging diseases, and AIDS is one of those. AIDS is one of the viruses that we definitely need a vaccine to. The problem is it goes into the body and begins to tweak immune cells, and it changes some of the functions of the immune cells, so that some of the activation processes, so that individual can't activate some of the cascades that we'll talk about as we get down the road in the adaptive system. But it's becoming more and more of a problem, and you guys are going to see something really cool. Scientists have already started taking viruses that cause disease. And since the 70s, 80s, we could tweak their genomes. We could make changes in their genomes. We could mess up proteins and see what it does to viral replication. We could do all kinds of things. And so scientists have started taking the AIDS virus and modifying it as a way to make a vaccine, but it's kind of an unusual vaccine. It make the virus that actually carries some modified genes that once it gets into the cells that it can infect, it actually prevents infection by the AIDS virus. So it's kind of a reverse vaccine in that it goes in and modifies and expresses some proteins down inside of the cell where the AIDS virus would normally want to be living and replicate, and the proteins that are expressed block the replication of the AIDS virus when we get in. We're using that for a lot of different systems. The model's already been used to specifically target some types of cancer cells, taking a virus and tweaking the receptors on the surface or the proteins that are on the surface of the virus and then targeting it toward other cells, and that we're beginning to do that in this approach too. So I imagine in the next 5-10 years you're going to end up seeing a vaccine that will come out that should be pretty effective against the AIDS virus as well. So that's the end of chapter 1.